Hello everyone, I'm Donna Roberts. I'm from the Medical University of South Carolina, and I don't see if speaker number one is still here. I'm one of those snooty hospital radiologists that he talked about. I'm a neuroradiologist actually, and before going to medical school I used to work at NASA headquarters, and back in the time when I was working at NASA headquarters, it was really exciting. A lot of work was being done concerning the changes of the human body in relation to space flight. Space Life Sciences Lab 1, SLS-1, was launched on the shuttle at that time, and we were learning a lot about the cardiovascular system, the musculoskeletal system, how to keep bones strong in space, muscles strong in space, about the importance of exercise and nutrition like we just heard from the other speaker. But I was always very surprised about why no one was really paying a lot of attention to the human brain in space. Let me see if I can. The slides aren't advancing. Great, thanks. And even today, very little is still known about the human brain. And so I put in a proposal to NASA to evaluate the brain imaging that was done of the astronauts before and after space flight. And my timing was pretty good because right about that time, there were reports from NASA astronauts who had spent long durations on the space station who were coming back with visual changes. And I think this was kind of touched on a little bit earlier. And when the astronauts got back onto Earth and had their pressures measured, there was increased pressure around their brains. And this was really first brought to light with this landmark paper by Matter et al., who reported on 7 astronauts with visual findings following 6 months in space. 5 of the astronauts had optic disc edema and globe flattening. And I have here a picture of an MRI of the orbit, which at the top is the normal orbit, and you can see how the back of the globe is rounded, very rounded, smooth surface. And on the picture below that, can you see my arrow? No. On the picture below that is the picture from Matter's paper showing that in this space station astronaut, the back of the globe is flattened, and it's very subtle, but I have it pointed out with the blue arrows, is that structure is the optic nerve, and it's where it attaches to the back of the globe, and it's very subtle, but you can see that it's swollen, and so that's optic disc edema. 5 of the astronauts also had other visual changes, including choroidal folds, cotton wool spots, and nerve fiber layer thickening. 6 had decreased near vision. When the measurements were performed to measure the pressure around the brain, it was found to be elevated. And you can see the pressures that were listed here in these 4 astronauts. Now the normal range is up to 20. So these were only mildly elevated. But the interesting part about it was in the 1 astronaut who underwent multiple repeated measurements, the pressure remained mildly elevated up to 19 months following spaceflight. And so NASA coined the term VIP syndrome, or visual impairment intracranial pressure syndrome, to describe these findings in the astronauts. Right now, the specific mechanism for the development of VIP, and it happens only in some of the astronauts, not all of them following long-term spaceflight, but the mechanism for its development is unknown. Matter in his paper put out 3 hypotheses. First, he suggested that microgravity fluid shifts could lead to a rise in the pressures in the veins in the head and neck, and that would increase pressure around the brain. He suggested that microgravity could interfere with the flow of fluid around the optic nerve, causing optic nerve injury, and also that altered pressure brought about by microgravity, or also by launch or landing, could change the pressure within the orbit and cause the findings. And so we're going to focus on his first hypothesis, which was microgravity fluid shifts leading to a rise in venous pressures in the neck. And it's common to see astronauts, such as astronaut Chris Hadfield here, on orbit showing enlargement of the veins of the head and neck. This enlargement of the veins reflects potential cerebral venous congestion, which could lead to impairment of the outflow of the fluid from the head. So the fluid is called the cerebral spinal fluid, and impairment of outflow of the cerebral spinal fluid would lead to an increase in pressure around the brain. And this is the only equation I have in here. But you can see that the resistance to CSF outflow and cerebral venous pressure contributes to increased intracranial pressure, which then can be transmitted to the back of the globe because the optic nerve is in continuity. The fluid space around the optic nerve is in continuity with the fluid around the brain. So we all know that it's very hard to study astronauts, particularly in that immediate post-flight time period. And so NASA is always looking for analogs to mimic the physiologic changes that occur to the body in space. And so we were very interested in looking at long-term head-down tilt bed rest as a suitable analog for studying the findings of VIP that we just discussed. So in head-down tilt bed rest, the head of the bed is tilted down 6 degrees, and normal subjects stay in bed for extended periods of time. They have to keep their head in this head-down tilt position at all times. And NASA provides entertainment and other duties to keep them occupied. But they're monitored 24/7 to ensure that their head remains in the head-down tilt position. Even while they're taking showers, it's kind of like a drive-through shower. And the only exception to when they can lift their head is when they're eating a meal 3 times a day for 30 minutes. They're allowed to lift up their head on one elbow, like you see this one participant doing, but other than that, they have to keep their head in the head-down tilt position the whole time. And so during bed rest, just as in microgravity, there is a shift of fluid from the feet and the chest area towards the head. And this shift in fluid stimulates central volume receptors in the heart, the carotid arteries, and the aorta. And that causes a diuresis, natriuresis, and a decrease in plasma volume and other metabolic changes that closely mimic those that occur in spaceflight. So that's why we thought that bed rest would be a good model for what's happening to the astronauts with the VIP syndrome. So we participated in this bed rest study. It was held at the Flight Analogs Facility at University of Texas Medical Branch, or UTMB, in Galveston. And we studied 8 normal subjects who were positioned in the 6 degrees head down tilt bed rest. They were maintained on a strict diet. And our objective was to evaluate for any potential structural changes of the human brain that occurred during bed rest. What we did was we acquired 3D MRI images of the brain before and after bed rest. So a baseline MRI scan was obtained. The subjects then were put to bed for 42 to 60 days. In actuality, the study was supposed to be 90 days long to mimic a 3-month mission upon the space station, but the study was being performed in Galveston, Texas, and it happened to be at a time when a hurricane came through, Hurricane Rita, so we had to evacuate some of the subjects early. So the images that I'm going to show you were from subjects who had undergone bed rest for 42 to 60 days. And at the end of that period, before the subjects were even allowed to get up out of the bed, they were taken to the MRI scanner, transferred from their 6 degrees head down tilt to the MRI table, and we obtained another MRI scan of their brain. And our study was the last study that they did before actually sitting up, so they were anxious to get their brain scan done. So the analyses that we did on these brain scans include the following. First, we looked for global volume changes of the brain. We also looked for whole brain shift. We evaluated any movement of the brain inside the skull, with the skull as the reference between pre and post bed rest. We looked for changes in volume of the fluid-filled spaces of the brain, which are called the ventricles. And we also looked for local brain tissue volume changes, which I will describe to you. One of the methods that we used in particular, because what can happen is if you take a brain scan of someone and then at another time repeat that brain scan, the person may have their head in a slightly different position than it was at the baseline scan, so we had to correct for that. And the way we did that was we used the skull as a reference. We extracted the skull from the pre and post bed rest images, aligned those, and used that as our alignment parameters to evaluate for brain shift. So here are our results. Following long-term head-down tilt bed rest, we found no global change in brain volume, and I'm going to show you examples of all these. But there was upward shift of the brain. The whole brain shifted upwards within the cranium. There was a change in the size of the fluid-filled spaces within the brain. And there were structural alterations involving important areas of the brain, which I'll also show you. So here's our first result. We really found no global change in brain volume in these long-term bed rest subjects. There was a change of approximately 1 to 2%, but this was not statistically significant. But what we did see was shift of the brain. We saw that the brain actually shifted upwards within the skull and rotated slightly backwards. And to help you get a better visualization of what this looked like, we took images of the brains of this is one particular subject where we had an image of the brain before they went to bed, and then another image of the brain after they went to bed. We extracted the skull, and we did a 3D reconstruction of the brain. And so we're alternating between the pre-bed rest image and the post-bed rest image. And so you can get a sense of how the brain actually shifted backwards and shifted upwards. And these findings are going to be a bit subtle. And unfortunately, my pointer doesn't work on the screen. But if you look at the top set of images before bed rest and after bed rest, these are images of the brain. These are slices of the brain going from side to side. So right down the middle of the brain, it's in the sagittal plane. And over towards the right is the nose, and to the left is the back of the brain. And I'm going to put red circles here. These are the frontal lobes. If you pay particular attention to the frontal lobes, the CSF spaces around the brain tissue of the frontal lobes is slightly more prominent after bed rest. I'm pointing out here a particular gyrus, or one of the foldings of the brain, a particular gyrus here, and with the yellow arrows, and here it is after bed rest. And these blue stars, I'm not sure if you can see, or blue asterisks, are pointing out the CSF space surrounding that piece of brain tissue, and it's slightly more prominent after bed rest. Down at the bottom, the two images on the bottom, before and after bed rest, show an area of the brain up at the top of the brain, up close to the skull. And the structure that I'm pointing out here in the blue arrows is one of the cortical veins, one of the veins that lie along the top of the brain. And before bed rest, it's surrounded by dark signal, which is the CSF. And then after bed rest, it's no longer surrounded by the CSF. Instead, the brain tissue is crowding against that vein and compressing that vein. And even though these findings are subtle, they have important clinical significance. And that is compression of the main venous outflow pathways of the brain can result in obstruction to the venous blood leaving the head. And venous outflow obstruction is considered the mechanism for the development of an earth-based medical condition called idiopathic intracranial hypertension that has been considered by some to be a model of VIP syndrome. So this was an important finding to see that these veins were being compressed after bed rest. Our third result was that the fluid-filled spaces, or the ventricles, were changing in size. I have here on this image, that's an axial image or transverse image through the brain. The red arrows are pointing out the ventricles. And so the ventricles changed in size during bed rest. In some subjects, interestingly, the ventricles got smaller. And again, it's going to be a subtle finding, but if you look at the image which is labeled A, on the left, I have a red arrow pointing out an area of the ventricle. And if you look at image B, this area is a little bit less prominent than it was in the image A. And in other individuals, the ventricles actually got bigger. So image C, you can see the baseline size of this subject's ventricles, and in image D, where the red arrow is, they're slightly larger. And these findings look very subtle if you just look at one image through the ventricles, but you've got to realize that these are 3D structures, and so it's the overall change that's important. And the fact that the ventricles are changing in size in these subjects who are normal, healthy, young subjects, it's very unusual for ventricles to even be changing in size in young subjects unless there's some underlying condition such as some medical condition or some medication that they're taking. So to show it maybe a little bit better, I don't know how well this is showing up on the screen, but I outlined in blue the ventricles, and then I'm switching back and forth between the pre-bed rest and the post-bed rest images, and you can see that before bed rest the ventricles are slightly smaller, and after bed rest they enlarge to the area of the blue outline. This is a subject where the ventricles got smaller. Again, these are movies showing the pre-bed rest image and the post-bed rest image in the same plane, just showing how the ventricles got smaller. Again, these are very subtle changes, but when we did the volumetric analysis, these changes accounted up to 10 to 22% change in ventricular size. And just to put that in perspective, a patient with Alzheimer's disease, ventricles would be expected to change approximately 5 to 16% in a year. And again, these were normal, young, healthy subjects. Another interesting thing about the change in ventricular volume was it correlated significantly with the amount of brain shift that we saw. So the amount of posterior rotation that we saw in the brains of these volunteers correlated with a change in their ventricular size. And that indicates to us that the brain shift resulted in an alteration of CSF homeostasis. And our final result was the finding of structural changes affecting widespread areas of the brain. They're indicated here in yellow are areas where we found increased tissue density, and blue areas are where we saw decreased tissue density. And the functional significance of these structural brain changes are unknown, but they definitely could be crucial given the fact that the areas where we are seeing all these structural brain changes are involved in adaptation to microgravity. So for example, for optimal performance in microgravity, astronauts have to learn how to reinterpret sensory inputs from the periphery. And a lot of that takes place in a part of the brain called the parietal lobes. Also, because the legs are used less for locomotion in microgravity, new motor control algorithms have to be developed by the brain, and that involves cortical plasticity in areas of the brain in the frontal lobe, such as the primary motor cortices and the supplemental motor areas, which are shown here in red. And then also, it's been demonstrated in astronauts changes in cognitive performance and changes in reaction time involving areas of the brain such as the frontal lobe. So the next steps are to evaluate the brain scans of the astronauts before and after spaceflight and see if similar changes, which we suspect would occur in the astronauts, to document whether or not those changes are occurring in the astronauts as in the long-term Bedrest subjects. We also want to get long-term follow-up imaging to determine if these changes remain or if they return to baseline. Unfortunately, the subjects were scanned at the end of Bedrest, but we have no long-term follow-up to see if these changes resolve, or if so, what the time course of resolution is. And then finally, we also recommend performing advanced MRI studies to assess. We've looked at the structural changes. Now we would also like to assess any physiological changes of the brain that could occur in response to these structural changes, such as changes in cerebral blood flow or brain tissue water in the Bedrest subjects and also potentially in the astronauts. And that's it. Thank you.
Are there any questions?
Which of the changes you have observed do you see as potentially most worrisome if they do not go back to original state in the long term? And do you see for prolonged space flight, do you see any potential remedies as to exercise positioning or other things? Are there any theories about how that might be dealt with?
Right. Yeah. I think one of the immediate issues is the issue with the seriousness of the issue with visual changes and increased pressure around the brain. And I think one of the potentially contributing factors to that is the brain shift resulting in compression of the venous structures along the top of the head. And so that's a very worrisome finding that we need to look at further. And thanks. And the issue is, do these changes remain? Do they resolve? We don't know. And if so, if they do resolve, is it just simply a matter of being back in Earth's gravity? Do we have to provide some type of centrifuge or something else if we're planning on long-term space flight? We just don't know.
Coming to your last point, did you observe a complete recovery after this test of all the anomalies?
Yeah, that's our biggest issue. We were not able to get the long-term follow-up brain imaging, so we have no idea. It's a big open question.
We'll do one more.
Two questions, really. Were you able to find any collaborative evidence of control group over the same amount of period which you viewed these people to see if there was natural variations in equilibrium? And the second one is there another group or any other research you could see where, let's say, divers who go through continuous changes in pressure, all right, showed similar type of effects?
Yeah, Thanks. I'm not aware of any similar type of studies being done in divers, but there is extensive literature out there showing that these changes are not seen in normal subjects otherwise. All studies, Alzheimer's studies, autism studies, they all include the control groups. And so there's a lot of standard literature out there. There's even studies that have been published comparing taking groups of subjects and imaging them on different scanners over different lengths of time, with or without hydration. You know, there's extensive literature out there showing that these type of changes do not occur in normal subjects.
Right. I think we have time for one more. Sorry.
Has anybody ever done any studies on people exposed to high gravity for a long period of time, such as the great mambo chicken?
Yeah, not that I'm aware of. Not these type of studies with the MRI scans obtained before and after hypergravity conditions.
All right, thank you.